Stem cells are undifferentiated biological cells that can differentiate into specialized cells, where they can continue to divide and act like a normal cell. Stem cells are usually found in multicellular organisms, such as humans, and come in two types, embryonic and adult stem cells. To be considered an official stem cell, it must have the properties of self-renewal and potency. Self-renewal is being able to go through cell division and stay undifferentiated, and potency is the ability to differentiate. First, we're going to focus on embryonic stem cells. The only way to harvest these stem cells is to derive them from the inner mass of a blastocyst. The embryo of a human becomes a blastocyst between 4 and 5 days after the fertilization period, and consists of anywhere from 50 to 150 cells. But the embryo does not come from the womb of the woman. Instead, it comes from an in vitro fertilization clinic, where the egg is fertilized outside of the body. Then they're grown through a process known as cell culture, meaning that cells are placed in a dish with culture medium and begin to divide. Cells don't always survive when they're placed on the culture medium, but if they do, they're soon gently scraped off and placed onto fresh dishes. This process, also called subculturing, happens repeatedly over a period of months, and each cycle is referred to as a passage. Once that cell line has been established, millions and millions of stem cells are generated. Cells that have not differentiated during this process are known as pluripotent, but later on they are stimulated to differentiate and specialize. Now we're going to talk about adult stem cells. An adult stem cell can be harvested in 4 different ways. It can be harvested through bone marrow, it can be harvested through extraction of adipose tissue through liposuction, it can be extracted through the filtration of blood, and it can be extracted from the blood of the umbilical cord. The origin of stem cells in certain tissues is still unclear, but scientists do have evidence to prove that stem cells are found in the brain, heart, gut, liver, bloodstream, skin, teeth, and numerous other places. The main role of adult stem cells is to maintain and repair the tissue that they're found in, so like when your skin scabs over after you cut yourself. Both types of stem cells have advantages and disadvantages for use in regenerative cell-based therapy. Embryonic stem cells are pluripotent, meaning that they can become whatever they're needed to be. However, it's thought that adult stem cells can really only divide and become whatever they were in the original tissue. Embryonic stem cells can easily be grown in culture medium, but adult stem cells, because they're much rarer in mature tissues, can't be grown as easily because there's a lot less of them. Stem cells have a plethora of potential uses. Studies of embryonic stem cells will show scientists information about complex processes that occur in the early stages of human development. Serious medical conditions can sometimes be the result of abnormal cell division and/or differentiation. Understanding how these processes can work can show the different ways a condition can arise and may suggest new ideas for treatment and therapy. They can also be used for drug testing through stem cell lines, but that's a little more complex as the conditions have to be identical on all stem cell lines, and they have to be stimulated to specialize into certain cells. The most important potential application, though, is the reparation and regeneration of cells and tissues for cell-based therapies. While people donate organs and tissues today, the demand is much greater than the supply. But since stem cells can be differentiated, it offers the possibility of a renewable source of replacement cells and tissues. With that, Alzheimer's, spinal injuries, and other debilitating diseases can be treated. Currently, there's much debate regarding stem cells. While adult stem cells are not controversial, embryonic stem cells are in the limelights of debates, because embryonic stem cell research includes the creation, usage, and destruction of a human embryo. To create a stem cell line, you're effectively destroying a human embryo, which is a pretty big issue, especially in the eyes of conservatives.